Last class we talked about how activation energy and the amount of activation energy affects how a chemical reaction happens, specifically how fast it happens. Lower activation energy is going to cause you to have a faster chemical reaction, whereas higher activation energy is going to cause you to have a slower chemical reaction. So that was just a review of chemical reaction basics, and we did that so that we could prepare ourselves for understanding the function, the structure and function of enzymes. So just to give you a little background information on enzymes, there are over 1,000 different types of enzymes in the human body. And enzymes are specific types of proteins. So before break, we talked about the 4 classes of macromolecules: carbohydrates, lipids, proteins, and nucleic acids. Enzymes, so all of these 1,000 different types of enzymes, are all examples of proteins. And they are important for biological reactions. Many people call enzymes biological catalysts, and catalysts are those molecules or substances that function in speeding up chemical reactions. So technically, enzymes are biological factors that speed up chemical reactions. Each enzyme is different because each enzyme, just like every other protein, has a different combination of 20 amino acids. So the different combination of amino acids is going to cause them to fold differently. And when they fold differently, they have different shapes. And when they cause these unique shapes, that also causes the enzyme to have a specific function in the human body that is different from any other protein or enzyme in the body. So enzymes are very specific, and the specificity is an important aspect of their function. So let's talk about a little bit about what enzymes actually do to chemical reactions. So again, catalysts are substances that speed up chemical reactions without being affected by the reactions themselves. Enzymes are biological catalysts that basically proteins that speed up the rate of reactions by decreasing, and this is the important part, by decreasing the activation energy. So let's take a look at our activation energy graph. So last time I likened a energy graph like this to rolling a rock up a hill. So let's say we've got to get our reactants AB to the top of this first hill in the red without the enzyme. It's going to take a lot of energy. So this is the energy used. You're taking a lot of energy to go from A to B to the top here in order to get to AB, which is your product. When you have an enzyme, the enzyme actually functions in lowering the activation energy. Now it does that by increasing the chance that A and B are going to be in close proximity to each other. So A and B are randomly bouncing around, like let's say a jar. And if you put an enzyme in there, the enzyme is going to allow A and B to come into closer contact with each other more often. By doing that, you need less energy in order to get A, B together. So what happens is your hill decreases in size. So it's a lot shorter of a hill than your first hill up here. And because of that, your reaction is going to happen much quicker. And the enzyme actually functions in speeding up this reaction. Now, this is extremely important for biological systems because a lot of the reactions that exist in our body will not happen without enzymes because it just takes far too much energy for them to happen naturally. So if there was not enzymes in our body, we would not have these reactions occurring, and then therefore we would not be living. So the enzymes actually play an important function in making reactions occur that normally wouldn't occur due to the high amount of activation energy that causes them to react into their products. So we look, if we look at this diagram here, you've got some frogs trying to jump over this activation energy barrier. It's a metaphorical barrier of rocks. And this one is very, very high. So it's going to take a long time for all these frogs to jump over in order to reach the side of the products. On the other hand, when you have an enzyme, what the enzyme does is it lowers that barrier by knocking off some of those rocks. And what that does is it allows the frogs to jump over a lot quicker. And you'll have a much faster product formation from your reactants. So this is with your enzyme, this is without your enzyme. So some important things about enzyme anatomy, and this is what we're going to be doing in the lab with our stop motion animations, is that you have two main things on an enzyme. And there's some other sites on an enzyme that will affect the enzyme's functioning, but these are the two main ones. You have a substrate molecule, which is the molecule that needs to undergo the chemical reaction. So this is really the important part of the reaction, is your substrate, because this is what's going to be caused or turned into your products. So it's the reactant molecule of the reaction that binds to the active site. And it is also the molecule, the reactant, that undergoes the chemical reaction. So your substrate molecule is going to be doing the changing. Now your active site is the other part that's very important here, because if you notice, each substrate molecule has a specific shape. Each active site is specifically designed, or it has its own specific shape that causes it to be specific for one substrate molecule, meaning that this enzyme is only meant to speed up the reaction of this particular substrate or reactant. Any other substrate will not fit or bind to this. It's like some people liken it to a puzzle piece. So puzzle pieces only fit into one specific puzzle piece. Or you could also talk about a lock and key. One key lock is going to fit only one key, and therefore it's very specific. So the active site is very much like the lock, and the substrate is very much like the key. So the actual reactions, or the actual site where the reactants undergo the chemical reaction, is your active site, and each active site fits only one specific substrate molecule. So let's talk a little bit about how enzymes work. Okay, in this particular diagram, your substrate, which is a dimer, okay, you got two molecules here, is going to enter the active site, which is specifically shaped for that dimer. Once the substrate enters the active site, the enzyme forms around the substrate, creating an enzyme-substrate complex. So what it means to be an enzyme-substrate complex is a combination of two things. Okay, it's a larger molecule or thing that is basically made up of more than one. So you've got the enzyme and you have the substrate, which forms this larger complex of molecules. Okay, so once that happens, a chemical reaction occurs in the enzyme that changes the reactant molecule into the product. Okay, so you've got the enzyme forming around the substrate here, and then you have a chemical reaction occur, which ends up breaking the bond between the dimer, and now you have two monomers released as your product. Okay, so the products are released, and the active site is now free to accept more substrate molecules. So if you look, once this is released, your active site goes back to normal, and another substrate can come in, react, and release. So this happens over and over and over again. So believe it or not, your enzyme is not used up during a chemical reaction. It can actually just basically, once it releases its products, it's going to go back and do the same thing over and over and over again. So enzymes are not used up after a single reaction. Instead, the same enzyme can catalyze hundreds of reactions. So we're going to talk about protein structure tomorrow, but for now what we're going to do is I'm going to show you a brief little video on some things that can affect enzyme function. Let's see here. Make that larger. Okay. So this is normal enzyme catalytic function, meaning that it's the normal way that enzymes speed up reactions. So if you look, in this you have your enzyme in blue, and you have your substrate molecule up here in green. And if you notice, your substrate molecule has a specific shape, and the active site is also specifically shaped. So what happens during the chemical reaction is the enzyme binds to its substrate. A chemical reaction is going to occur that digests this. So this is a digestive enzyme. So the chemical reaction occurred, digests it into two products, and it continues to do so over and over and over again. So again, it can happen multiple times. So something that can affect enzyme function is something called an enzyme inhibitor. And an inhibitor there's two types of inhibitors, a competitive inhibitor and a non-competitive inhibitor. So I'm going to show you what a competitive inhibitor does to enzyme function. So a competitive blocker is a molecule that may not be exactly like your substrate, but has a similar shape. So this particular inhibitor doesn't have the full shape of the substrate, but it has partial shape of the substrate. So it binds to that part of the active site. And by doing so, when the substrate molecule comes in, the substrate molecule cannot bind. So when the inhibitor is in the active site, your substrate cannot bind and the chemical reaction will not occur. The other type of inhibition or inhibitor is called a non-competitive enzyme inhibitor. And this is a little bit different. Okay, so it does not compete with the substrate for the active site. Instead, it does something else to the enzyme, which we'll see in a second. So here you have the binding of a non-competitive blocker at a different site on the enzyme. When that binds, you actually cause a conformational or shape change or folding pattern change in the enzyme, in the protein of the enzyme. And when you do that, your active site changes shape. So you don't have anything actually in the active site, it just changes the active site shape because the protein folds differently when you have inhibitors. So when that happens, your substrate tries to come in, but it can't fit into the active site, so no reaction occurs. And so just like the competitive blocker, your enzyme does not work properly. So that is our little tutorial on enzyme function and how that affects chemical reactions. See you in class.